Hello students, today we will discuss for the Krebs cycle and this is called as the TCA cycle. TCA cycle means tricarboxylic acid cycle. This Krebs cycle, it is named after the scientist Hans Adolf Krebs. In 1937 he proposed this cycle and he worked on the oxygen consumption of pigeon breast muscle. Now this cycle it occurs in the mitochondrial matrix, and matrix is having the necessary enzymes for the reactions. Come to the process of glycolysis, which we have discussed in the previous video. Glycolysis is a pathway in which the glucose it gets reduced to form 2-pyruvate. This pyruvic acid it comes to the matrix of the mitochondria with the help of the proteins, and here this pyruvate it gets combined with the coenzyme A to form acetyl coenzyme A, and in this process NADH it is also produced. Now the enzyme which is responsible for the reaction, that is pyruvate dehydrogenase. This reaction it is considered as a link reaction. This is considered as a link reaction because it links the glycolysis with the Krebs cycle. So it is a gateway step. Now proceed with the cycle. Oxaloacetic acid it combines with this CoA coenzyme A to form citrate. Here citrate it gets converted into cis aconitate, which converted into isocitrate. Isocitrate it gets converted into oxalosuccinate. Oxalosuccinate it gets converted into alpha keto glutarate. It is converted into succinyl coenzyme A, which is converted into succinic acid or succinate. Succinate it gets converted into fumarate. This fumarate it gets converted into L-malate or malic acid, and finally the malate it gets converted into oxaloacetic acid. So this is what the cycle is, which is called as the TCA cycle. Now here we will see the functioning of the enzyme, which enzyme it is responsible for the reaction. This oxaloacetic acid it combines with CoA to form citrate, and enzyme is citrate synthase. Here citrate, it gets converted into cis aconitate, and in this process a water molecule it is removed. This is known as dehydration. Cis aconitate, it combines with the water molecule to form isocitrate. This is called as hydration, and for both the reactions the one enzyme it is responsible, that is called as aconitase. So you can write here that aconitase, it works for the two reactions, and in one what happened, the dehydration occurs where the cis aconitate it is formed, and after that the hydration occurs where isocitrate it is formed. Now come to the next reaction, and here isocitrate it gets converted into oxalosuccinate, and the enzyme is isocitrate dehydrogenase. In short, it is written in this way. Here the NADH it is formed. So you can write this like this, and actually the isocitrate it gets converted into oxalosuccinate. Here the dehydrogenation it occurs, and after the formation of oxalosuccinate the decarboxylation occurs, and the enzyme is decarboxylase, where the CO2 it is evolved, and after the decarboxylation of oxalosuccinate what is formed? Alpha ketoglutarate it is formed. Now when the alpha ketoglutarate it is formed, after that the dehydrogenation and decarboxylation both process occurs, and the product which is formed that combines with coenzyme A to form succinyl coenzyme A, and the enzyme which is responsible, that is alpha ketoglutarate dehydrogenase. So here you can write the production of NADH, and you can write the evolution of CO2 as well. So here the succinyl coenzyme A it is formed, the enzyme that we have seen alpha ketoglutarate dehydrogenase. Now succinyl coenzyme A it transfers its high energy bond to the GDP. GDP is what? Guanosine diphosphate, and after the transfer of the high energy bond GDP it gets converted into GTP. So this is what the substrate level phosphorylation. This is called as substrate level phosphorylation, because here the metabolites they are responsible for the formation of high energy molecule. After the formation of this GTP, in a coupled reaction ATP it is formed. How? With the help of enzyme phosphokinase. The GTP it gets converted into ATP. Actually the phosphate of GTP it is transferred to ADP, and so that ATP it is formed. That is why called as in a coupled reaction. First, the GTP is formed. From the GDP, when the high energy bond of succinyl coenzyme A, it gets transferred to GDP. GTP is formed, and in a coupled reaction, the ATP is formed when the enzyme phosphokinase makes the transfer of phosphate to ATP, and ATP it is formed. Now, here the enzyme which is responsible for the conversion of succinyl coenzyme A to succinate, that you can write as succinate thiokinase. This succinate, it gets converted into fumarate through the enzyme succinate dehydrogenase, and here FADH2 it is formed. So you can write in this way. FAD it is converted into FADH2 because of the dehydrogenation. It is the only reaction in the citric acid cycle which involves the direct transfer of hydrogen from the substrate to FAD without the participation of NAD. Now this fumarate, it gets converted into malate, the enzyme that is fumarase, and here the hydration occurs. One molecule of water it is added to form malic acid or malate. This is called as malate, or you can write as malic acid. Malate, it is dehydrogenated into oxaloacetate or oxaloacetic acid, right? And you can write the enzyme as malate dehydrogenase, MDH. In short, you can write in this way. So these are the enzymes which works, and you can see for each and every reaction the different enzymes they are there. So we have discussed in detail this TCA cycle. This is the diagram which you can see. If you want to recall that we have discussed, here the citrate it gets converted into cis aconitate through the enzyme aconitase, and here first of all we have to consider the combination of oxaloacetic acid with coenzyme A. This is a condensation reaction to form citrate, and here the enzyme citrate synthase it is responsible. After that cis aconitase it is formed where the water molecule it is removed, dehydration occurs, and here after the formation of cis aconitate, hydration occurs where the water molecule it is added to form isocitrate. For both the reaction, same enzyme it works. Isocitrate it gets converted into oxalosuccinate with the help of isocitrate dehydrogenase. In this step, NADH it is formed. Oxalosuccinate it gets decarboxylated into alpha ketoglutarate with the help of enzyme decarboxylase. Here the CO2 it is evolved. Alpha ketoglutarate it gets dehydrogenated and decarboxylated to form succinyl coenzyme A, and actually after the dehydrogenation and the decarboxylation, the product it combines with coenzyme A to form succinyl coenzyme A. The enzyme which is responsible, that is called as alpha ketoglutarate dehydrogenase. This succinyl coenzyme A gets converted into succinate, the enzyme that is succinyl coenzyme A synthase, and here the GDP it is formed. you know, in the conversion of succinyl coenzyme A to succinate, GTP is formed where the high energy bond of succinyl coenzyme A it is combined with GDP to give GTP, and in a coupled reaction ATP is formed when the phosphate it gets transferred to ADP. So you can see the conversion of ADP into ATP and the enzyme that is responsible, phosphokinase. Here in the conversion of alpha ketoglutarate into succinyl coenzyme A, you can write for the formation of NADH plus H, and also the CO2 evolution takes place. So this is substrate level phosphorylation, which you can see when the succinyl coenzyme A gets converted into succinate. Now this succinate it gets converted into fumarate with the help of succinate dehydrogenase, where FADH2 it is formed from FAD. One molecule of water it is added to fumarate to give malate or malic acid, the enzyme that is fumarase, and malate it gets dehydrogenated to form oxaloacetate, and the enzyme is malate dehydrogenase. So we have revised as well this TCA cycle. Again, your queries they are always welcome, and you can have the interactive live sessions with me. You can solve your doubts, post your requirements, we'll make video for you, and now we will meet in the next video. Bye for now.